Martha. In this short video we'll look at higher key area 2, the replication of DNA. So again, these are the course notes we'll start with. We need to know first of all the DNA replication happens prior to cell division. So if you remember cell division from National 5, this is because both daughter cells maintain the chromosome complement and are still diploid after mitosis. This can only happen if the DNA is doubled before cell division, and that's where DNA replication comes in. The enzyme which replicates the DNA is called DNA polymerase. So our enzymes end with A's quite often, and their name associates with their job. So this one makes DNA, so it's called DNA polymerase. DNA polymerase cannot replicate the DNA until a primer joins. So we're going to look at the role of primers, which are a short strand of nucleotides which bind to the free end of our template strand. So you need to know your structure of DNA before you can learn how to replicate DNA. A nucleotide is your basic unit of DNA, so a primer is made up, single-stranded, short strand of these nucleotides. We need to know that the free end of the DNA is the end that starts with the deoxyribose sugar. We'll work our way through the stages, but we're going to talk about DNA unwinding and unzipping by breaking the hydrogen bonds that join the two opposite strands together. And then we can talk about why DNA replication is different for both strands. That one can be replicated continuously, whereas one can only be replicated in short fragments. Okay, so already we talked about that it happens prior to cell division. We need to have a DNA molecule to copy. We need to have free DNA nucleotides. Free means they're floating around inside the nucleus, because remember these are the building blocks of DNA, so we need to have these to build a new strand. Primers are required before the replication process can start, and there's two different enzymes involved in this, and you'll need to learn the names of both of them. So our first enzyme is DNA polymerase. This is the DNA the enzyme which replicates the DNA. DNA polymerase requires a primer before it can start replication. So said a primer is a short strand of nucleotides. These always bind to the free end of the template DNA strand. Once the primer is added, this allows the DNA polymerase to start adding and joining free DNA nucleotides to create a new complementary copy of the template strand. Okay, so here we can see the DNA. First of all, to make a copy of it, we have to open up the molecule. So the first thing you should always start with is by saying the DNA unwinds, so you're untwisting the double helix, but then it also has to unzip to expose the bases. Unzipping it means breaking the hydrogen bonds between the bases. This gives us a top strand in blue and a bottom strand shown in red. We call this fork the replication fork, and the DNA molecule is going to keep unwinding and unzipping from left to right, so the fork will move from left to right. So that means we have one direction of replication. That's important. The strand that runs 3 to 5, i.e. the strand that starts with the deoxyribose sugar, we call this strand the leading strand. Replication takes place from 3 to 5, so this one can just keep being replicated. As the DNA molecule unwinds and unzips, the DNA polymerase, once it's started, can just keep running in that direction and continuously copy that strand until the whole DNA molecule has been copied. However, the other strand runs 5 to 3. This is the opposite direction that DNA polymerase works in. Therefore, because the lagging strand is anti-parallel, it cannot be replicated continuously. As you move left to right and the DNA unwinds and unzips, you keep getting a new 3 end being exposed again and again and again as the molecule unzips. So DNA polymerase has to keep replicating this in short fragments as the new 3 end is exposed each time at that replication fork. So let's look at the leading strand first of all, because that's the easy one to understand. So we've exposed the bases, there's an exposed 3 end there, so our primer attaches to that 3 end. The primer is important as it allows the DNA polymerase to then start adding and joining together these complementary free nucleotides, and this creates a new strand of DNA. As we said before, the DNA polymerase is working from 3 to 5. So because the new strand it's making is anti-parallel, you can say that DNA polymerase always adds nucleotides to the 3' end of the new strand being made. And because of this, it works in a continuous fashion on the leading strand, following the replication fork, left to right, up the molecule. Okay, so DNA polymerase can only add DNA nucleotides in one direction. That's the key thing. It can only run from 3 to 5 because it always adds the free nucleotides to the 3' end of the new strand being replicated. And you can see that in this picture here. On the left, the primer has bound to the 3' end, which means the DNA polymerase, shown as the green circle in the middle picture, can then start. In the middle picture, it's already joined the first half dozen bases. Okay, it's adding and joining these nucleotides to form that blue, newly synthesized strand of DNA. And the picture on the right shows that as the DNA molecule unwinds and unzips further, the DNA polymerase can just keep going in that direction, adding and joining new DNA nucleotides to the 3' end of the new strand which it's synthesizing. And that goes on and on and on. So the leading strand is replicated continuously. Okay, so now we look at the lagging strand. The lagging strand runs from 5 to 3. Now, if DNA polymerase could replicate in both directions, it can simply start at the 5' end and also replicate the strand continuously. But it cannot. DNA polymerase can only replicate in one direction, which is 3 to 5. So, what that means is, as this DNA molecule unwinds and unzips, you keep getting another 3' end exposed. On this picture here, the purple section on the right is the first primer to join. Because the bottom is the 5' end, that primer has joined to the 3' end when that first section was unzipped and exposed. So when that purple primer has joined there, the DNA polymerase has replicated down the way from the 3 to the 5. Now the easiest way to explain this in an SQL question is just to memorise the text on the left. 2 parts. One, you say the lagging strand is anti-parallel. The second part is you say that DNA polymerase can only replicate in one direction. Both these pieces of information together explain why it is replicated in fragments, as otherwise it's quite hard to word. So we've replicated our first section. Now let's look at what happens next. So, as it's unwound and unzipped up the way, it's exposed a new 3' end. So this time a red primer has joined to the exposed 3' end. And again, DNA polymerase can now replicate the section between the primers. So it's replicating down the way again, remember, because it always replicates in one direction only. So we're getting a second section made, or a second fragment made. Then, you guessed it, the third section has opened up. There's a new 3' end again. So again, a red primer attaches to the newly exposed 3' end, and DNA polymerase can now replicate by adding and joining 3 DNA nucleotides together and adding them to the 3' end of the new DNA strand. So now we've got 3 different fragments. This fork is going to unwind and unzip further. So again, at the very top of the page where it's still bonded, you have a GGTC. That is going to become exposed next, and our primer will have to bind to that, so the next fragment can be replicated, and so on and so on. So the leading strand is easy, it's just continuous. The lagging strand is a bit more complex. Replication happens in the same way, it's the same enzyme, DNA polymerase. It still requires primers to bind to the exposed 3' end before it can happen, but because it can only replicate in one direction, we're having to replicate in short fragments. So finally, when you have all these different fragments, they have to be joined together. So that's when our second enzyme comes in. And DNA ligase's role is to join the DNA fragments together. Okay, so a simple sentence to memorize. DNA ligase joins the fragments of DNA together on the lagging strand. And it's only the lagging strand ligase is involved in. And this is what it would look like. That top one, the original template's in blue, and it's been replicated continuously. So it's getting a new red strand made. On the bottom strand, that one starts with a phosphate, making it the 5 to 3 direction, making it anti-parallel. So therefore, it's replicating right to left, as that's the direction that DNA polymerase replicates in, and it's going to keep being replicated in fragments until the DNA molecule has been completely copied, and these fragments have been joined together by DNA ligase. So, these are the most common questions you'll get. What happens before DNA can be replicated? You'd always say that a primer must bind, and you must say it must bind to the 3' end of the DNA template. And that's required before DNA polymerase can begin to replicate. There's 2 enzymes involved, both end with A's, DNA polymerase and DNA ligase. DNA polymerase is the enzyme which is going to replicate the DNA, so it's involved in both strands. DNA ligase is only involved in the lagging strand because its function is to join the fragments of DNA together. If you're asked to explain why the leading strand can be replicated continuously, simply say that it runs from the 3 to 5 direction, and the DNA polymerase replicates the template by adding 3 nucleotides to the 3' end of the new strand. So this means by saying that it's also running from 3 to 5. If you're asked to explain why the lagging strand is replicated, remember both halves. First of all, you say it's anti-parallel, so you're talking about how it's structurally different, and then you say DNA polymerase can only replicate in one direction. So that's why it's replicated in fragments.